Well, my dear students, today part 3 cell cycle. In the part 3, we are going to discuss different type of phases in the interphase. In the previous class, I already explained you what is meant by interphase and what are the different phases in the interphase. In the different phases, these are G1 phase, G2 phase, S phase, right? G1 phase, S phase, G2 phase and G0 phase are the different phases in the interphase. Coming to the G1 phase, it is called post-mitotic phase. The meaning of post-mitotic means after the completion of mitosis, the proceeding step is called as G1 phase. That's why we call it as post-mitotic phase. It is also called as pre-DNA synthesis phase. The meaning of that means before synthesizing the DNA replication, the phase is called G1 phase. That's why it is called as pre-DNA synthesis phase. Okay. Now coming to this G1 phase, it is an interval between mitosis and the initiation of DNA replication. That means the stage between the mitosis and the stage between the synthesis of DNA. In between this stage, the G1 phase is present. That's why it is called as an interval between the mitosis and DNA replication phase. Now coming to the second phase, dear students, in this phase, all the cell organelles. What are the cell organelles? In a eukaryotic cell, there are both double membrane cell organelles are there and single membrane cell organelles are there. Double membrane cell organelles, but the duplication of this double membrane cell organelles takes place in G2 phase, not in G1 phase. Whereas rest of the all single membrane cell organelles are going to be synthesized in the G1 phase only. If you come to this, the cell size increases maximum. Why the cell size increases maximum? Due to the increased protoplasmic content, increased RNA, increased different type of proteins which make the cell size maximum in the G1 phase, right? And also in this phase, I already said that the protoplasm content had increased. Why the protoplasmic content had increased? Due to the increased RNA and increased synthesis of proteins in the cytoplasm of the cell. Next proceeded after this, next proceeded to S phase. Actually, dear students, this point will be applicable to S phase only. Why I have written here, in the competitive exams, you will get a question, in which phase of the cell cycle do the replication of cytoplasmic DNA takes place. So, most people will get confused that replication of cytoplasmic DNA will take place in S phase. No, it is not, exactly not. The replication of cytoplasmic DNA takes place in G1 phase only. So what is meant by cytoplasmic DNA? Dear students, the DNA present in mitochondria and chloroplast is called as cytoplasmic DNA, right? So these cell organelles are present in the cytoplasm. That's why this DNA is called as cytoplasmic DNA. You understand? Now coming to the S phase. S phase is called as DNA synthesis phase. In this, so it is most important point, it is an invisible phase of M phase. So in the M phase, it is said to be an invisible phase. Why? Because the DNA replication starts in the S phase only. That's why it is called as invisible phase of mitosis. Right? Next is the replication of nuclear DNA takes place in this phase along with histone protein synthesis. Dear students, histone proteins are the basic proteins which are associated with the eukaryotic DNA to form a nucleosome complex. These histone proteins are entirely absent in prokaryotes. So coming to here, histone proteins are also synthesized in the S phase only. It's very important point. One more point here, most of the students get confused here is, there is no change in the chromosome number of S phase. Only the DNA content is going to be double. So what is the purpose of doubling of the DNA content? Because whatever the DNA content present in the parent has to be equally distributed to the daughter cells. In order to fulfill this phenomena, the S phase, which is a phase where the DNA content is going to be double. Understand? So this double DNA is going to be shared equally to the daughter cells. So here, for example, DNA content is initially 2C, while the completion of S phase, it is going to become 4C. You understand? Now, no change in the chromosome number takes place, and the most important point here, centrioles are going to be duplicated in the late S phase. What are centrioles? Centrioles are amembranous cell organelles which are present in animals only, entirely absent in plants. Centrioles take part in the formation of spindle fibers. You understand? So centrioles will duplicate in the late phase of S phase. And where do the centrioles duplicate? Cytoplasm of the cell. So centrioles will duplicate in the cytoplasm of the cell. These are self-duplicating cell organelles, and whereas mitochondrial DNA and chloroplast DNA will be synthesized in the G1 phase, whereas these are called as cytoplasmic inheritance. What is meant by cytoplasmic inheritance? What are the genes which are inherited from cytoplasm of the parent cell to the progeny, offspring, that is called as cytoplasmic inheritance. So finally, I conclude that S phase is the synthetic phase. The DNA replication takes place. DNA content is going to be double here. No change in the chromosomes. Duplication of centrioles will be takes place. Okay. The next class we will discuss about G2 phase and G0 phase. Thank you.